For 30 years, the prevailing consensus in neurodegenerative research has treated amyloid-β as a pathological intrusion, a toxic byproduct of precursor protein cleavage that accumulates in the aging brain. Yet the amyloid precursor protein is ancient. It is deeply conserved across evolutionary history, with functional homologs found in nematodes and flies, as well as mammals. A molecule of that antiquity, expressed at high levels in every neuron and released in direct response to synaptic activity, is unlikely to exist solely to cause a disease of late life. Zhen Huang proposed an inversion of this model, framing amyloid-β as a physiological competition signal that arbitrates which synapses survive activity-dependent competition. In this view, Alzheimer's is not an external intrusion, but a normal homeostatic pruning system that has lost its balance. If the peptide performs a required physiological function, then pure clearance strategies are biologically miscalibrated. To see if this framework holds, we can look to a 2026 assessment that cross-referenced Huang's theory against several years of independent empirical data. This requires a distinction between a theory of disease onset and a theory of molecular function. Claims about how a disease starts are difficult to prove in postmortem tissue, but claims about what a molecule is for can be tested through direct intervention. To survive, the framework must hold up across 4 distinct arenas: loss-of-function genetics, in vitro microglial pathways, CSF biomarkers, and clinical trial readouts. Validating the molecule's physiological grammar is a prerequisite for modeling its pathological failure. The foundational logic of Huang's framework relies on a single observation: amyloid-beta exhibits strictly concentration-dependent duality. At low concentrations, between 100 to 200 picomolar, the peptide acts as a trophic signal, enhancing long-term potentiation and increasing the probability of synaptic vesicle release. Endogenous monomeric amyloid-beta is required for normal plasticity. Depleting it induces synaptic failure and memory impairment. But at high concentrations, typically in the nanomolar range, the peptide forms oligomers that suppress exocytosis and deplete the recycling vesicle pool. Huang uses the bacterial lantibiotic nisin as a template for this behavior. Nisin solves the problem of local competition by acting as a protective monomer near the secreting cell, while forming toxic pores that kill neighbors at a distance. Mapping this onto the mammalian synapse, the active terminal secretes amyloid-beta to protect itself, while generating a distal oligomeric halo that marks less active neighbors for removal. A single concentration gradient can theoretically generate 2 opposite activities, transforming a diffusible peptide into a spatially selective competition signal. To test the model's predictive power, we have to look at the genetic consequences of removing the punishment signal machinery. While a pure toxicity model expects healthy tissue upon signal removal, a competition model expects disinhibited, exuberant growth of structures that should have been pruned. This second outcome is what appears in the somatosensory cortex of precursor protein knockout models. Following whisker plucking, these neurons failed to retract their axonal terminals during the expected rewiring process. A sparse deletion control, where the protein was removed only from the retracting axons, reproduced the failure. This established that the defect is cell-autonomous to the axon that should have lost the competition. The high-affinity receptor implicated in this punishment signal is PirB, or LILRB2 in humans. In ocular dominance plasticity, monocular deprivation in PirB mutants leads to an exaggerated growth of the losing ipsilateral input. In amyloid overexpressing models, where oligomers are elevated, the visual cortex exhibits hyperpruning, a shrinkage that is entirely prevented by deleting the PirB receptor. The presence of unpruned axonal exuberance matches what we expect when a competitive signal is withdrawn, confirming the existence of this architecture in vivo. One major gap in the original framework was how monomeric amyloid-beta specifically restrains the immune cells, the microglia, responsible for the pruning. A 2024 study supplied a pathway showing that monomeric amyloid-beta binds the precursor protein, signaling through a chaperone called RIC8A to suppress the release of matrix metalloproteinase 9. Deleting this pathway in vivo produced neuronal ectopia caused by excessive basement membrane degradation, a phenotype that was rescued by pharmacologically inhibiting the matrix protease. However, the microglial suppression required monomer concentrations of 50 to 500 nanomolar. This is a geometric paradox. The lower bound of this microglial effect is already at the threshold where amyloid-beta begins to suppress synaptic transmission. It suggests that the concentration axis described in the bacterial analogy does not smoothly create a safe local gradient. The switch between protective and toxic states seems to be driven by molecular conformation, monomer versus oligomer, rather than a unified concentration axis. Without a concentration-driven gradient, the theory must rely on a different selectivity mechanism, such as exposed phosphatidylserine on the membrane, to target specific synapses. This conformation-driven model received support from an independent direction, human biomarker data published by Espace Group in 2025. Their data showed that higher levels of cerebrospinal soluble A-beta-42 track consistently with normal cognition, regardless of the total amount of plaque in the brain. In fact, the relationship between soluble peptide levels and cognitive health was stronger than the relationship between total plaque burden and dementia. This was confirmed longitudinally in genetic mutation carriers, where the amount of residual soluble peptide was a better predictor of clinical progression than the PET amyloid burden. Under this model, the formation of plaques acts as a physical sink, consuming the functional soluble pool of monomers. This monomer depletion disinhibits microglial cytokine release, which in turn drives tau pathology and further amyloid production. The data supports the view of Alzheimer's as a proteinopenia, where the loss of a protective soluble species matters more than the accumulation of an aggregate. These theoretical predictions were put to the test by the final readouts of the major Phase 3 clinical trials. The framework predicted that any drug that binds and removes the functional monomer would fail, even if administered very early in the disease. Solanezumab and crenezumab, both of which target the soluble monomer, failed to show benefit in preclinical and presymptomatic genetic cohorts. In contrast, agents like lecanemab and donanemab, which are designed to spare the monomer while removing aggregated species, achieved a real slowing of cognitive decline. This success validates the principle of protecting the soluble species while clearing with deposits. The theory also explains the results of the INVOKE-2 trial, which tested an antibody called AL002, designed to increase microglial activity. The trial failed to show clinical improvement, despite confirming that it had successfully hit its target and activated the immune cells. The failure of microglial activation corresponds with the theory's central premise, that chronic microglial activity, disinhibited by monomer loss, is the core lesion rather than the solution. Friction points remain, such as the discovery of complement proteins competing for the same receptors, and human tissue work showing tau oligomers tagging removed synapses. However, the structural claim remains resilient. Amyloid-beta is an innate immune and competitive signaling molecule, not a biological error. Its pathogenesis is the catastrophic failure of the protective half of an ancient physiological loop. The synaptic competition framework leaves us with a single, enduring therapeutic principle. To protect the brain, do not remove the monomer.